So what is preclinical? So basically preclinical means before giving to humans. That's all the thing is. So you need to study about the pharmacology, toxicological aspects in laboratory animals. Okay. So when you do that, for example, this particular drug has improved this particular condition in animals. So you have the proof of your concept. Okay. So based on this proof of concept, you can develop a hypothesis. Okay. So to develop a hypothesis, this preclinical study is very, very important. Okay. Phase 1 clinical trials. The question to be answered in phase 1 is the treatment safe? That's all it is. Okay, whether it's treating the patient, whether it's curing their disease, that's all is immaterial. Am I giving a toxic substance to my patient? Is this really safe? That's the first question to be answered. Okay, usually includes a very small number of people, even fewer than 100. Okay, people with specific disease or conditions or even healthy volunteers are involved in this. Goal is to learn the maximum tolerated dose with acceptable side effects. Okay, safety is carefully checked at each dose before approving further increases. Okay, you will be completely monitored, like vital signs, blood tests, ECGs, and other medical tests are done frequently to detect any adverse effects because of this drug itself. Okay, drug induced effects. Researchers learn how the experimental treatment is taken up by the body, moves through the body, it's broken down into various metabolites, and how does it exit from the body. Okay, this is nothing else pharmacokinetics. Okay, this is otherwise called as pharmacokinetic studies. Okay, any interaction with food or other medications taken together is also studied. Okay, all of this information is crucial for designing safe protocols for the next testing phases. The ultimate goal of phase 1 is to determine safety. Okay, and what dose is safe. Okay, maximum dose which is safe for healthy volunteers or specific group of patients who have that condition. Okay, so safety, safe dosage range that can be used in phase 2 to start assessing efficacy. Okay, efficiency is definitely checked in phase 2 only, not in phase 1. Understand this. If any safety concerns is observed, then halt the trial. Phase 2 clinical trials check for efficacy and dosing. Okay, basically the question is, does the treatment work? It includes more participants than the phase 1, up to even several hundred people. Of course, you're going to monitor safety along with your treatment outcome, along with your adverse effects also. Okay, in phase 2 trial, participants who have a specific disease or condition get the studied treatment at a dosage level found to be safe in phase 1. Okay, so in phase 2 there is no normal subjects. Okay, in phase 2 it's only the patients, because you are going to check the efficacy, right? Then the research team monitors the participants' health and disease symptoms to get a initial gauge of efficacy. For example, how does the efficacy is for the 10 mg of the drug? Then you can slowly increase it to 20 mg. Then check how is the efficacy, what are the side effects, how is the tolerance, all these things. For example, you can have various biomarkers also. For example, the tumor size may be measured in cancer drug trials, or the viral load for antiviral medications. So I'm giving X amount of drug, the viral load has decreased to 10%. Can I give 20% more of the drug so that the viral load decreases further with acceptable side effects? All these things comes under phase 2 trials. Okay, you're going to check the efficacy and dosage for that particular efficacy with what are the side effects. Okay, this is a phase 2 clinical trial. So clinical lab tests also analyze specific other genes, proteins, substances like biomarkers that can indicate the experimental treatment's effect, right? Like improving in lactate or improving in other biomarkers like PCT sort of things, right? In phase 2 clinical trials, researchers refine the methods to set a standard or goal and optimal procedures for large phase 3 trials. For example, the plan is to bring down the lactate from 10 to 2, or bring down the lactate to less than 2, okay, or bring down the PC to less than 0.5, or decrease the tumor size to so and so. All these goals can be set, because that will be the maximum achieved in your phase 2, right? By giving this amount of drug, this amount of benefits has been achieved in phase 2, okay? Does that reflect in a larger population in phase 3? So that questions should be framed in phase 2 only, okay? So the phase 2 clinical trials tells researchers more about how safe the experiment is, because you're conducting on a larger group compared to phase 1, phase 2 is a larger group, and how well it works before advancing to even more a larger group, that is phase 3.